This episode of Cell and Gene, the podcast, is brought to you in partnership with Thermo Fisher Scientific. Thermo Fisher's cell therapy processing instruments are designed to help customers transition from process development to commercial manufacturing, utilized as standalone devices or integrated as part of a closed modular process. Thermo Fisher Scientific recommends Gibco CTS DynaSelect Magnetic Separation System, which is a next-gen cell isolation and activation instrument. Gibco CTS Xenon Electroporation System allows customers full control to optimize for a variety of cell types and payloads. And Gibco CTS Rotea Counterflow Centrifugation System is a closed cell processing system supporting a broad range of protocols for cell separation, washing, and concentration. Customers can rely on and streamline their drug development process with Applied Biosystems QualTrack qPCR and dPCR quality control tools for robust and reliable genetic analysis across various phases of drug development, supported by relevant, compliant documentation. Hello, listeners, and welcome to this episode of Cell and Gene, the podcast. I'm happy to introduce my only repeat guest to date, someone who needs no introduction, Dr. Peter Marks. Dr. Marks is a hematologist-oncologist serving as the director of the Center for Biologics Evaluation and Research within the Food and Drug Administration. He joined the FDA in 2012 as Deputy Center Director for CBER and became Center Director in 2016. Dr. Marks, welcome back to Cell and Gene, the podcast. This is such a treat, not only for me, but also for our listeners. So happy to have you here.
It's really a pleasure to be here. Thanks so much.
You're absolutely welcome. So, all right, let's jump right in. All right, we are here to talk about base editing specifically. And so I wanted to hopefully have you level set the conversation of, where we are with base editing in terms of cell and gene therapy. It's unmet medical need. What is its potential? What excites you most about base editing, and also what concerns you most about base editing? So it's kind of a mouthful, but let's start at the top.
So thanks very much. So I think we can extend the discussion today to base editing and perhaps prime editing, which is a version of base editing that extends it a little bit. But the base editing is a technology which in some way it kind of makes working with DNA, it's kind of similar to how you would be editing a Word document, the idea that you can go in and take a single letter and change it. Because of that, the ability to alter just one nucleotide, one piece of the genetic code very accurately, there are potentially thousands of diseases that might be addressed where you just have a single base pair that is off. So I think there's a lot of potential here, and that's obviously for these for rare genetic diseases. If you then expand base editing to include prime editing, and prime editors basically take what base editing does and expand it slightly by using a more complex enzyme combined with a more complex guide to be able to edit small stretches of DNA. They can address an even broader array of genetic defects. And depending on what you're dealing with, one might be preferable over the other. But once again, this is a way of potentially dealing with an incredibly wide array of genetic defects and correcting them. And so incredibly exciting because of the number of different diseases that might be addressed. A little bit scary still because we don't understand everything about the potential off-target effects of these technologies. That is, it's wonderful to be able to potentially fix these genetic defects. The hope is, though, that we don't introduce others because whenever you're using a genetic sequence to match up there is that possibility that you can match up in the wrong place and create unintended effects. Now, so far, with base editing technology, people have been looking at this, and at least in when it's done in the cellular level, it looks like the number of correct cuts versus the number of incorrects is still very favorable. But I think we'll still have to see how that goes over the course of time, and there's still much more to be learned here. Because not only do we want to be able to do this in cells taken outside of the body and then put back into it, but potentially the real excitement comes from being able to potentially administer a base editor or a prime editor and have that happen in the body without having to go through the extra step of taking cells out and giving them back. The advantage of taking cells out is that after you've done the editing, you can actually do sequencing to make sure you haven't introduced something that's problematic. On the other hand, that adds a lot of complexity and potentially expense to the whole process. So really an exciting area that we'll learn more about. I mean, when you think about it, the rate at which these developments have occurred, they remind me of Moore's law. We've just seen, we had genome editing technologies previously with meganucleases and TALENs, and then we had CRISPR-Cas9. Then we had the base editors. Now we have prime editors, and people are starting to riff off of those. So tremendous amount of progress. And hopefully our ability to make sure that we're not seeing off-target effects keeps up with that so that we can really move this field forward.
Yeah, good. Okay, thank you for that. And thank you for including prime editing too. We've covered both on cell and gene. And so I'm glad that we brought that into the discussion too. Perhaps to talk a little bit more about the challenges or the bigger challenges that biotechs are facing, focused on both base and prime editing, whether it be off-target effects, what have you. When it comes to those challenges, since we're kind of early on here, are there potential solutions? Can you talk us through that a little bit?
Yeah, well, I think the issue of assessing this issue for off-target effects is one that there are enough people working on that that I think those will be addressed by scientific advancements over the next years, because we're already addressing some of them. I worry less about some of making progress there than some of the basic stuff, like being able to make these in a cost-effective manner with high quality and be able to have them studied effectively. I think we probably are going to need to come up with some new playbooks for how we figure out what crosses the finish line to an approved products for these genome editing therapies, because they will be, almost by definition, very targeted therapies to potentially small groups of individuals. Some may be for larger groups, and understanding how you manufacture them, study them, and get them across the finish line will be something that I think will be important. And I think for biotechs, what I think they worry about most, at least from having been on the other side, is uncertainty, right? So I think that to the extent that we in what we do as regulators can start to help to provide some guidance in this area, it will hopefully be helpful. So potential solution, obviously, Obviously, we want to see the science continue to advance so that we can understand off-target effects better. The manufacturing technologies advance so that we can bring down the cost of manufacturing. And obviously, then from our perspective as regulators, the potential solution is that we can hopefully get some guidance out there that essentially outlines the process of how you go about bringing these forward so that they can cross the finish line into approved products.
Yeah, sure. And I think maybe that's, the guidance that you and your team are working on to kind of help push this forward. Maybe that's the best way to drum up the next question, because I want to talk a little bit about, from a regulatory perspective, what are you and your team keeping your eyes on? Because, everyone under the umbrella of the biotech or, the sponsor company, they really care about what's happening from a regulatory perspective. And so when it comes to this genome editing, all of it, what are you and your team focused on from a regulatory perspective?
Yeah. It's from the regulatory perspective, it's obviously the safety of these products that will ultimately be front and foremost. I think the obviously we also care about effectiveness, but we have a reasonable sense of that from the field and from where things are. Better understanding and thoroughly understanding the issues around safety are really important. And one of the problems here is we really don't have a way of doing the equivalent of accelerated stability for a drug, which is that you would like to be able to very rapidly know whether there are going to be genomic alterations that are going to cause problems in the future. Now, in the cellular case, you can get at that when you take cells out, do this, and you can potentially do sequencing to get to a fair amount of that. But the concern here is that we want to make sure that there aren't going to be problems that come up years from the initial treatment that cause problems. So that is probably front and center as we think about this. Obviously, as this potentially migrates into being a directly administered type of treatment, we'll want to think even more carefully about off-target effects or potential off-target effects. So that to me is kind of front and center here, because otherwise base editing and prime editing potentially get around a lot of the other problems with some of the vector gene therapies that we have, because this is in and out, and the correction is made as opposed to needing to have persistence of the vector itself.
Sure. Okay, that makes sense. I want to talk a little bit about, within cell and gene therapy in the sector as a whole, there are some clinical holds going on. And so what do the recent clinical holds say about the FDA's views on gene editing?
I think I can just talk, I'll speak in general about this, which is that I think we've recently had some transitions in our office at FDA in terms of a transition to the Office of Therapeutic Products, a greater focus on the gene therapy, chemistry, manufacturing controls, and on the clinical trials in this area. I think what we may be seeing right now is we are in a transition period from a period where we were somewhat conservative about what we were letting move forward to a place where we will be a bit more permissive and will continue to follow the science in allowing things to be moving ahead once we are comfortable that the risks are adequately controlled. So I don't think I would read too much into what we have done in the past, but be assured that we're going to do our best to follow the science here, and allow things to move forward when we are comfortable that the risks involved have been adequately taken into account.
Sure, of course. Okay, thanks. That's helpful. While we're on the topic of clinical holds, I would just like to hear what you have to say just about them overall and in general, kind of broadly. What typically needs to happen for both potency assays and manufacturing quality control for a hold to be released anyway?
Well, when we place something on clinical hold, it's usually because either there's a quality issue, or there's a safety issue. It's very rare that something is put on hold because there's concerns about effectiveness. So it's either the product quality or product safety. We'll keep safety aside for now, because the issue there would be that we think that there's something that is concerning in terms of adverse events that are occurring. So for quality, the reason why we would put something on hold for quality is because we are concerned that the product is either not being manufactured consistently, that there are contaminants that we're concerned about that haven't been controlled, or that there's not adequate definition of the product, among other things. And so I think in general, when we see holds for quality, that tends to be things. And I guess I neglected to say the other reason why you might see a hold is that in the pediatric setting, you don't have linkage with some type of data from perhaps a non-clinical model that you could have prospect of direct clinical benefits. So those are the kinds of things that can get there. But in terms of manufacturing, what gets us is that we need to be able to see that, particularly as products advance, but we like to make sure that products will be made in a manner that are consistent, well characterized, ideally with potency assays. But we are as our guidance that we have issued in the past and will continue, you'll see in the future. I think we've had kind of this long standing policy that early on in development, we're willing to accept some more leeway in a product's specifications than as it gets closer to development. That is it's a twin-edged sword. On the one hand, it allows people to proceed more rapidly early in development. On the other hand, for some of these products that make it through development very quickly, it means that sometimes the process development has not occurred adequately, and so things get slowed down at the end. So, long story short, we need to, for something to come off hold, we have to make sure that we feel confident that there will not be a quality issue that is going to affect product safety.
Mm-hmm. Yeah, fair. Okay. So one more question as we've been talking. I'm curious about whether CBER considers an inheritable gene edit a deal breaker for approval or not. And so my question is the FDA prepared to approve a base editing therapy that alters the germline and becomes inheritable?
So I think I want to peel that apart into two questions. The first is that can we tolerate some uncertainty about administration of a base editor or prime editor that in doing its job could possibly have an effect on the germline as an incidental effect. In other words, that's not where it's targeted, but that as one of the issues that could occur by treating either a young individual or for that matter, somebody where you get some correction of this in a germ cell, and that is heritable. I think that kind of possibility is one that we can possibly accept here moving forward as something that it's possible, but it doesn't mean that you can't use base editors or prime editors in women of childbearing potential. I think that that doesn't have to be a place where things get stopped. And I think we'll want to make sure that there's the smallest chance of that happening. But I don't think that is going to be a deal breaker at the end of the day. I'll take so that's the one question, the kind of the use of prime or base editing in individuals, and this slight possibility that there could be germline correction. I think we can accept that as something that we're looking to try to mitigate it as much as possible, but that shouldn't preclude the treatment of individuals with base or prime editors. Then there's the issue of actually deliberately correcting heritable conditions. That currently is not permitted by our appropriations law. So we're not allowed to accept an application for review at FDA if the product is going to create a heritable genetic modification. That's something that's an appropriations rider on FDA, and that's the law that we abide by. Obviously, we continue to follow developments in other places in the world where some heritable genetic modifications are being permitted, but in the United States, in humans specifically, heritable genetic modifications are not permitted at this time. And so if that changes at some point, we'll be ready for it. But for right now, that's the state of things in the United States.
Great, fair. Thank you very much. All right, we are at the end of the episode, and as my listeners know, at the end of every episode, I ask my guests a question to get to know who they are when they're not in the office or the lab. And last time we talked, Dr. Marks, we talked about your passion for sailing. And as I understand it, I'm not a sailor myself, but as I understand it, as with most things in life, when it comes to sailing, timing is everything. So what I'd love to hear from you for our audience would be maybe one or two tips for the listeners, what they need to know about the basic elements of sailing. I assume timing is one of them, but what do our potential sailors out there need to know if they wanted to get started, say today?
If they want to get started today, first of all, when you start to go out to sail for the first time, go out on a very nice day when there's not a lot of wind, and go out and basically don't worry too much about all of the details, because there is so much theory that you can know. If you go out on a day where there's not too much wind, 5 to 10 miles an hour of wind, you can kind of get the sense for what is going on sailing with a small amount of instruction to learn how to tack and to jibe a boat. But the major piece of that is go out on a day that's a nice day when there's not too much wind. It might seem a little boring, but much easier to start to learn to sail when there's not an overwhelming amount of wind. Start small and build.
There you go.
Maybe a little bit like science.
I think there's a connection there of why you're so passionate about the two. Well, that makes sense. And I think, certainly you don't want to be on choppy waters either. So that's great. All right, listeners, that wraps up this episode of Cell and Gene, the podcast featuring CBER's Dr. Peter Marks. Dr. Peter Marks, thank you so much sincerely for your time and insight. This has been such a pleasure. I really appreciate it.
My pleasure. Thanks very much.
Listeners, be sure to go back and listen to episode 40 of Cell and Gene, the podcast, my first interview with Dr. Peter Marks. I promise you that is well worth your time as well. And we'll talk to you soon.